Down syndrome and all that stuff, and there was a book written called The World of Nigel Hunt, with a little Down syndrome boy, and Dr. Russell was British, and so was Nigel Hunt. So he was all excited about this book, and he'd be lecturing on genetics, and he'd get this thing of spittle that sort of accumulate in the corner of his mouth, and he'd just keep going, and we're like waiting for it to drip. It was kind of gross. Last month, I get my alumni magazine, and Dr. Russell just retired.
Oh, God.
I thought he was 100 when I had him. How many years ago was that? 30 years ago or more? How is that possible? But sure enough, that was him in the picture. I mean, he didn't really look that different. So I don't know how he did it, but anyway, I forgot. Oh, so I remember still stuff that Dr. Russell said. So for me, sense strand and anti-sense strand made sense. The sense strand is the one that you copy into RNA, because there's less chance there's going to be a mistake in it. If this is a copy of that, you could have made a copying error. So when you make your proteins, you're supposed to make them off your best copy. Now your textbook has different figures that kind of confuse me. I don't know what strand they're talking about, so we're not really going to worry about it too much. But I do remember Dr. Russell saying the sense strand is always the one that you copy. In fact, he used to give us homework problems, and the homework problem would be something like this. And it would go on forever and ever and ever. Then it would probably have a codon up here that was codon for start, and codon down here that was for stop. And then he'd ask us, if we were going to build a complementary strand of DNA, what would the sequence of letters look like? And so we have to go G, T, A, C, T, C, G, oops, I put a U, C, what was it? C, A, T, C. And then he'd say, okay. G. Thank you. I told you I make mistakes sometimes because I rush. Okay, and then he would say, okay, for the next part of your homework problem, if the cell, we're going to do protein synthesis, not copy the entire genome, but do protein synthesis for just this one gene, what would the RNA look like, the mRNA?
Like an anti-strand, right?
Right. So it would be C would be complemented by a G, A by, this time, U. Because remember, in RNA nucleotides, there's no T's, there's U's instead. So it would be GU, A, T would still be complemented by A, G by C, U, C, G, U, G. Did I get that right? So that would be your mRNA strand. And then he would say, okay, now I want you to go to the table in your book and look up what codons these are for. Who's got a textbook with them? Good. Go to chapter 7. I forget what page. There's a table of codons, mRNA codons. It's in my PowerPoint, but I didn't plug it in because I didn't want to have to put the screen down. What page is that? 208. Okay, page 208. Okay, Cole's got the table. There's literally a table in your book, page 208, that tells you the amino acid for every different codon. We know these things. So what's the codon for GUA? What's the amino acid that GUA codes for?
Valine.
Valine. You recognize valine as an amino acid? What does CUC code for? Leucine? And what's GUG code for? I think it might be valine again.
Yeah.
So, if I were building a protein from a gene, my protein would be 3 amino acids long. It would be a tripeptide. Those are the kind of homework problems they used to give us, only they were much longer, so after a while you were so sick of copying letters over, it was ridiculous. In the books, they always list the codons by mRNA language, not by DNA language. The reason is because the cell makes proteins by reading the mRNA copy. Okay. My great-grandma dies, passes down the family cookbook to my grandma. Eventually my grandma dies. The cookbook goes to my mom. My mom dies. The cookbook goes to who? I have a sister. We got a problem now. My sister wants the cookbook. I want the cookbook. What are we going to do? Make a copy. And to be fair, I have to take half of the book as a copy and half of the original book. And my sister has to take the other half of the copy and the other half of the original book. That's DNA replication. When you're making 2 daughter cells, you got to do semi-conservative replication, right? So each daughter cell gets some copy. That's fair. Okay? I cherish this family cookbook. It's got all my favorite recipes in there. Great-grandma's, I forget already, great-grandma's date nut bread, and grandma's spice cake recipe, and my dad calls me up one day and he's like, "So are you coming out to visit this weekend, Jude?" I'm like, "Yeah, all right, I'll come." So I gotta drive all the way out to freaking Sebring. "How about you make that spice cake of your grandmother's and bring it with you?" "Really, Dad?" "You're the one who's retired. You like to cook in your spare time. I'm busy, I'm working." "Oh, whatever, I'll make the damn cake." So I bring it out to him, he's like, "It's a little dry." So I want another scotch with it. Anyway, so I gotta make the spice cake. I cherish this cookbook, so I don't keep it in the kitchen. I keep it in my office under a glass case. And when I want to get a recipe out of there, I take it out of the glass case and I make a photocopy of the recipe. So I can save the book without ruining it. The book goes back in the glass case. I take the copy out to the kitchen to read it and to make the spice cake from it. That's what mRNA is. It's the message. mRNA stands for messenger RNA. I don't want to synthesize proteins off my DNA because I don't want to ruin it. If I have kids, I got to pass it on to my kids. I don't want to ruin it. It's my best copy I got. So instead, I make a photocopy. That's what mRNA is. It's a working copy of the DNA. Did I copy the entire cookbook? No. If my dad had asked me, "Well, when you bring the spice cake, I also want cheesecake, I want brownies," I said, "No, we're not doing this. One recipe at a time." Now, theoretically, I'm going to show it to you on the board, one recipe at a time, one gene at a time. But if you think about a complex organism, even something as complex as a bacterial cell, they're probably going to be synthesizing multiple proteins at the same time. You don't, for dinner, just have rice and nothing else, unless you're really poor. You probably have rice, and you have beans, and you have, steak, and you have salad, so you have multiple things. So, when you do protein synthesis, the cell copies the DNA for one gene at a time, directly. For the gene that codes for the needed protein. And mRNA is a single-stranded copy. Why do I have to make a double-stranded copy? I'm not passing it on to the next generation. It's just my working copy. So it would be wasteful of me. I'd be wasting nucleotides, wasting enzymes, wasting time, wasting energy. I don't need a double-stranded copy. I only need a single-stranded copy. So RNA is your single-stranded copy. Okie dokie. Before we go further, got a lot of other issues to talk about. And don't let me forget, you're going to need an RNA shopping list also. See how much better I do when I don't try to follow notes and PowerPoints? It's like, I just kind of meander. I never do the same thing 2 semesters in a row. I don't know how I got from point A to point B, but as long as I got there. And then fortunately, I'll have somebody like Holden tell me when I forget to mention something. Like every once in a while, I'm like, why are all these cats rubbing on my legs? Oh, I forgot to feed them. All right, I just need a color change for my own enjoyment. Okay, we're going to call this differences between DNA and RNA. Hi. Okay, let's put DNA over here. DNA stands for deoxyribonucleic acid. Because DNA is 2 strands of nucleotides, and remember a nucleotide is a sugar, which is a little ribose sugar, phosphate, nitrogen-containing base or nucleotide base or whatever you want to call it. So the ribose stands for the sugar, and the sugar is deoxy. It's missing an oxygen. What's RNA? Good. This one is